Hello dear students, this is Dr. Shabana Begum, Associate Professor from the Department of Zoology, Maharani Science College for Women, Bangalore. I am going to speak to you about the histological structure of mammalian kidney pertaining to BSc Zoology 4th semester, Unit 5. The content of this session would be to know the histology of kidney, the various structures of the uriniferous tubule, then histological details of different parts of the tubule, and also juxtaglomerular apparatus. The learning objective, you can see, so at the end of the lesson, the students will be able to learn and understand the histological details of different parts of the kidney along with their functional aspects. Before explaining the histological structure of the kidney, let me give you a small gist about its external anatomy as well as its location. Fine. So you can mention kidneys are bean-shaped, reddish-brown structures acting as chief excretory organs. Fine. They are retroperitoneal in position, that is, they lie between the dorsal body wall and the parietal peritoneum in the superior lumbar region. They extend approximately from the level of the 12th thoracic vertebra to the 3rd lumbar vertebra. Thus, the kidneys receive some protection from the lower part of the rib cage. An adult's kidney weighs about 150 grams, that is 5 ounces, and its average dimensions are 12 centimeter long, 6 centimeter wide, and 3 centimeter thick. And even say that the size is about the large bar of soap. The lateral surface of the kidneys is convex. Fine, it is convex. Then after mentioning about the size and weight, we can always mention that these are highly vascular organs and receive 25% of cardiac output. Approximately 1200 ml of blood each the kidneys receive for the process of filtration per minute. And the kidney produces urine by ultrafiltration of blood, which is then modified by selective reabsorption and the specific secretion by cells of uriniferous tubules, which are the 3 different steps during the urine formation. Fine. The medial surface of the kidney is concave and has a vertical cleft called the renal hilus that leads into a space within the kidney called the renal sinus. Several structures including the ureter, the renal blood vessels, lymphatics and nerves enter or exit the kidney at the hilus and occupy the sinus. 3 layers of supportive tissue surround each kidney. The fibrous renal capsule adheres directly to the kidney surface. It provides a strong barrier that prevents infection in surrounding regions from spreading to the kidneys. The middle layer is a fatty mass called the adipose capsule, which helps attach the kidney to the posterior body wall and cushions it against blows. And the outermost layer is the renal fascia, which is a dense fibrous connective tissue. Fine. So when you take a section through a kidney, it reveals 3 distinct regions: the cortex, the medulla, and the pelvis. It is about the internal anatomy. In a few words I am going to explain. The most superficial region, the renal cortex, is light in color and has a granular appearance. Deep the cortex is the darker reddish-brown renal medulla, which exhibits cone-shaped tissue masses called medullary or renal pyramids. The broad base of each pyramid faces toward the cortex and its apex or papilla points internally. The pyramids appear striped because they are formed almost entirely of roughly parallel bundles of microscopic urine collecting tubules. The renal columns, that is the inward extensions of cortical tissue, separate the pyramids. Each medullary pyramid and its adjacent cap of cortical tissue constitutes a lobe of the kidney. That is one of the approximately 8, there will be 8 in fact within the kidney, 8 lobes will be there. One of them is composed of the medullary pyramid and its adjacent cap of cortical tissue. Fine. So the lateral to the hilus within the renal sinus is the renal pelvis, a flat funnel shaped tube which is continuous with the ureter leaving the hilus. Branching extensions of the pelvis form 2 or 3 major calyces, each of which subdivides to form several minor calyces. Cup-shaped areas that enclose papillae of the pyramids. That means these are the cup-shaped areas that enclose papillae of the pyramids. The calyces collect urine which drains continuously from the papillae and empty it into the renal pelvis. The urine then flows through the renal pelvis and into the ureter, which transports it to the bladder to be stored. In fact, the walls of the calyces, pelvis and ureter contain smooth muscle which contracts rhythmically and propels urine along its course by peristalsis. Fine. So once again, in this particular slide, you can see the internal anatomy of the kidney, which I have just explained. There is an area which is called as hilus, with a notch-like appearance, in which the renal vessel, nerves and the ureter enter or leave the kidney. There is a covering of capsule which is thick and is made up of collagenous fibers, smooth muscle fibers. Fine. Apart from that, I even said that the kidneys as such, you can say, is composed of the 2 main regions, the outer cortex and the inner medulla region. There are structures like renal pyramids which are seen clearly here. The apex of the will be facing the renal sinus and their bases will face the cortex region. Apart from that, the renal calyces are clearly seen. The major calyces and the minor calyces can be seen in this particular slide. Apart from which, you can mention the kidneys as such are made up of collection, that means a million uriniferous tubules, numerous uriniferous tubules we mention, and each uriniferous tubule is composed of nephron and the collecting duct. So these uriniferous tubules are accompanied by capillary network called vasa recta that runs parallel with them, and these represent vascular part of countercurrent exchange system that regulates the concentration of urine. So next is this, you can mention about the uriniferous tubules. These are the structural and functional units of kidneys, which are also called as actually nephron. Then the uriniferous tubule, I said, is composed of nephrons and the collecting ducts. And when you speak about the nephron comprises of the renal corpuscle, proximal tubule, then Henle's lobe, distal tubule, etc., along with which in the uriniferous tubules collecting ducts are also included. You can give the explanation like this once again. Each kidney contains over 1 million tiny blood processing units called nephrons, which carry out the processes that form urine. In addition to nephrons, there are thousands of collecting ducts, each of which collects urine from several nephrons and conveys it to the renal pelvis, which you have just studied about. And each nephron consists of glomerulus, this is ball of yarn we say, which is a tuft of capillaries associated with renal tubule. The end of the renal tubule, called the glomerular or Bowman's capsule, is blind, enlarged and cup-shaped and completely it completely surrounds the glomerulus, which is much as a well-worn basketball glove encloses a ball. Some information. Then collectively the glomerular capsule and enclosed glomerulus are called the renal corpuscle. The glomerular endothelium is fenestrated, that is, it is penetrated by many pores, which makes these capillaries exceptionally porous. They allow large amounts of solute-rich, virtually protein-free fluid to pass from the blood into the glomerular capsule. This plasma-derived fluid or filtrate is the raw material that is processed by the renal tubules to form urine. The external parietal layer of the glomerular capsule is a simple squamous epithelium. This layer simply contributes to the structure of the capsule and plays no part in forming the filtrate, while the visceral layer, which clings to the glomerulus, consists of highly modified branching epithelial cells called podocytes. These are also called as foot cells. The branches of the octopus-like podocytes terminate in foot processes or pedicels, which intertwine with one another as they cling to the basement membrane of the glomerulus. The clefts or openings between the foot processes, called filtration slits or slit pores, allow the filtrate to pass to the interior of the glomerular capsule, a region which is called as capsular space. This is all about the Malfugian corpuscle, which you can see in the slide 2. The Malfugian corpuscle, also called as renal corpuscle, comprises of a double-walled Bowman's capsule along with the glomerulus. And the remainder of the renal tubule, which is about 3 centimeter long and has 3 named parts, such as the proximal convoluted tubule, which makes a hairpin loop called the loop of Henle, and then winds and twists again as the distal convoluted tubule before it empties its products into a collecting duct. The collecting ducts, each of which receives urine from many nephrons, run through the medullary pyramids and give them their striped appearance. So this slide will show you the various regions of the nephron along with the collecting ducts, which form together the uriniferous tubules. The diagrammatic representation of the nephron is very clearly seen. We can explain again in the same manner which I just gave you the explanation about. The expanded portion of the nephron is called as the Bowman's capsule, right, which continues as the proximal convoluted tubule, then the loop of Henle, distal convoluted tubule, which finally joins the collecting duct. This is yet another representation of the uriniferous tubules, which are named according to the course they take, that is convoluted or straight, with their location, that's proximal or distal, their wall thickness, whether they are thick walled or thin walled. Then, as you all know, this proximal convoluted tubule, it originates from the urinary pole of corpuscle and located in the cortex. Proximal straight tubule is also known as thick descending loop of Henle, which descends into medulla, and then there is a thin descending limb that makes hairpin turn and returns towards cortex. There is a thick ascending limb that known as the distal straight tubule. It ascends through medulla into the cortex. So now this slide will show you the outline histological detail of kidney, which designates as the 2 regions as the outer cortex and the inner medulla region. The cortex region is composed of the Malfugian corpuscles, proximal convoluted tubule and distal convoluted tubule, and sectional view. Apart from that, the outer zone of medulla comprises of Henle's loop and collecting tubule. Inner zone contains the thin segment of Henle's loop, collecting ducts, and ducts of Bellini, also called as papillary duct of Bellini. I have already given you the explanation about the renal corpuscle. Then you can just have a look at this histological structure, which is stained in the hematoxylin and eosin differential staining. here in the first diagram will give you the complete view of the cellular arrangement of the kidney and this nephron. The two regions that cortex and medulla are seen clearly. There are also the region called renal pelvis and also medullary papillae. The second diagram will give you the structures maybe at 40x of renal corpuscle in the cortex region. Also there are sections of proximal convoluted tubule and distal convoluted tubule, while in case of medulla region one can find the sections of loop of Henle and a part of collecting tubule or ducts. Next, this particular slide will show you the structure of the renal corpuscle, also called as Malfugition corpuscle, where you can find right in the center of the diagram the glomerulus, which is the tuft of blood capillaries, and the surrounding double-walled cup-shaped Bowman's capsule. Fine. It has two layers actually. The inner layer is called visceral layer, which contains cells called podocytes, which I mentioned, and they extend processes around the glomerular capillaries. And the outer layer or wall is called the parietal layer. It's also called a somatic layer, and its wall is lined by means of simple squamous epithelium. This slide once again show the structure in a sort of a section of the region of Bowman's corpuscle or glomerular corpuscle, where one can find the afferent arteriole entering into the Bowman's capsule and forming a tuft of itself, and then leaving the glomerular region the form of efferent arteriole. About this one more explanation, you can say the renal artery, which enters into the kidney, divide to form interlobular arterioles in the cortex, and these give rise to afferent arterioles which open into glomerulus. Fine. The capillaries that form the glomerulus receive an afferent arteriole and feed into efferent arteriole, which leaves the renal corpuscle. And these efferent arterioles in the outer part of the cortex open into the second capillary network, the first being the glomerular network, and which is called as Peri-tubular capillaries, which surround the convoluted tubules of the cortex. Fine. So this you can not see, but the explanation maybe later when you have your offline classes, we will explain about it once again. Fine. So this glomerular capillaries are lined by means of endothelium, is highly fenestrated. I have mentioned these endothelial cells which line these capillaries has large number of pores called as aquaporin 1 water channels that allows fast movement of water through the epithelium. Then this glomerular basement membrane shows cells which are principal component of filtration barrier, and this is a prominent, very prominent in section stain with PAS, that is periodic acid skiff stain, which can be seen only with the particular stain. So this a sectional view of histological structure of the cortex region especially is seen clearly here. It is taken as of the magnification of 40x. It's still a low bar only. So you can mention here there are sections of the glomerulus with the capillaries having red blood corpuscles. Surrounding the glomerular region is the Bowman's capsule region. The space can be seen clearly there. There is a pole which is called as urinary pole. From that pole region itself, the proximal convoluted usually arises, and this is just the sectional view. That's why it's called as histological structure. Right? So you can mention about the presence of the different sectional view of the proximal convoluted tubule, then distal convoluted tubule, and there are podocytes mentioned here. There are cells called as mesangial cells, about which I am going to explain later, and there are also cells which are called as juxtaglomerular cells. Fine, this much about this particular slide. And then next is, I just said I will mention about mesangial cells. Actually, these are specialized cells which are present in the glomerulus attached to the capillaries. These cells with their extracellular matrix is called mesangium. It functions as macrophages in intraglomerular regions, and they phagocytose material that accumulates on glomerular filters. They give structural support to the podocytes there. Then they secrete IL-1, that is interleukin 1, and platelet-derived growth factor. So the diagram shows you the position of mesangium and mesangial cells, which are drawn in the green color. The rest of it shows the presence of the afferent arteriole, efferent arteriole, Bowman's capsule, podocytes are seen clearly, then proximal tubule region. There are also glomerular capillaries as mentioned in this particular slide, which are enclosed within the Bowman's capsule. The mesangial cells actually regulate glomerular blood flow. Some cells are located outside the corpuscle along the vascular pole, and these cells are known as lattice cells. They are flat cells with fine cytoplasmic processes, and they form part of juxtaglomerular apparatus. Very important here, from 1 mark they may ask a question. This controls glomerular blood flow. Okay, in fact, this are called as JG cells, juxtaglomerular cells of this apparatus. They produce a hormone called renin. Fine. About this I'll tell you later. One about functions of it, which can be seen later. The next, then what are podocytes? This slide will give you a very clear picture of the structural diagrammatic representation of podocytes, right? Podocytes are modified cells to form stellate cells, and the primary branches of these podocyte cells give rise to secondary branches, which are called as pedicels. And these pedicels rest on the capillary endothelium. Between two pedicels, or adjacent pedicels, there are small pores known as filtration slates, and they, in fact, these arrangement helps in filtration of blood. That is the first step in urine formation. And these podocytes are usually present in the visceral layer of Bowman's capsule. Now coming back to the sectional view of the various tubular structures of the nephron. So you can mention each region of the renal tubule has a unique cellular anatomy, which reflects its specific role in processing filtrate. Right? The first cells of the tubular region, that is that of proximal convoluted tubule, we have to explain. These tube-like structure, which is convoluted, is lined by means of some say they are pyramidal or low columnar or cuboidal cells with a large spherical nucleus, and these actively reabsorb substances from the filtrate and also play a role in secreting substances into it. They have large mitochondria, and their luminal or exposed surfaces bear dense microvilli that tremendously increase their surface area. This so-called brush border dramatically increases their capacity for reabsorbing water and solutes from the filtrate. So you can mention the proximal convoluted tubule is lined by cuboidal cells with large spherical and basal nucleus. The distal free ends of the cell bear microvilli that form brush borders, hence called brush bordered cells. This is a characteristic feature of proximal convoluted tubule cells. This makes their lumen appear narrow. And the cells also show infolding of the basal cell membrane of the cells, forming compartments where mitochondria and polyribosomes are found, yet another distinctive feature of cells lining the proximal convoluted tubule. So you can just read the labelings which are mentioned in the slide. And the second part of it is actually the sectional view of the loop of Henle, where the thin limbs of the Henle is lined by means of simple squamous epithelium, while the thick limbs are lined by means of cuboidal epithelium, and there is no brush border, fine, lining the lumen of these cells. So in this particular slide, actually the sectional view of medulla region is shown here, hence you will not find the sections of glomerular region or Bowman's capsule or even proximal convoluted tubule and distal convoluted tubule, but you can find the sections of the thin limbs of loop of Henle, thick limbs of loop of Henle, and you can even find the capillaries, right, plus ducts which are called as papillary ducts. Then there are also straight ascending limb of loop of Henle. Then there are also connective tissue stroma seen here and there in this particular section. And I have already mentioned that the U-shaped loop of Henle has descending and ascending limbs. The proximal part of the descending limb is continuous with the proximal tubule and its cells are similar. Similar means they are cuboidal, and the rest of the descending limb is called the thin segment. It is lined by means of simple squamous epithelium that is freely permeable to water. So the epithelium once again becomes cuboidal or even low columnar in the ascending part of loop of Henle, which therefore becomes the thick segment. In some nephrons, the thin segment is found only in the descending limb of the loop of Henle. In others, it extends even into the ascending limb as well. Next region is the distal convoluted tubule. It is lined by means of columnar epithelial cells without brush border. There are few microvilli seen here, very few. In fact, the cytoplasm is acidic and the lumen appears larger. Fine. So you can explain the cells of the distal convoluted tubule, like those of proximal convoluted tubule, are confined to the cortex and are cuboidal. They are, you can say, low columnar, but they are somewhat thinner and almost entirely lack microvilli. In fact, their structural features reveal that these tubules play a greater role in secreting solutes into the filtrate than in reabsorbing substances from it. So you can show this particular slide of collecting tubule also is mentioned here, both the diagrammatic representation of distal convoluted tubule, which is the figure drawn on top, and that of collecting tubule is shown below. Then you can mention the collecting tubule and duct of Bellini. In fact, they are lined by means of simple columnar epithelial cells with well-defined cell boundaries. You can see the beautiful nucleus placed right in the middle of the cells. The nucleus is large, prominent, and the cytoplasm is clear. These are concerned with selective reabsorption, active secretion, and transport of urine. There are two types of cells seen in collecting tubules and collecting duct. These are called as the light cells and the dark cells. So the light cells are also called as collecting duct cells, CD cells, also known as principal cells of the system, which are pale staining, and they have basal enfoldings and also a single primary cilium in them. Few short microvilli. They have a small spherical mitochondria present, and they possess abundance of vasopressin, also known as ADH, antidiuretic hormone, regulated water channels, that is AQP, aquaporin 2. which is responsible for water permeability of collecting duct. While the dark cells, also known as intercalated cells or icy cells, are lesser in number and their cytoplasm is denser and they have many mitochondria and no basal enfoldings. Basally, they show interdigitations with neighboring cells and they have numerous vesicles present in apical cytoplasm. The function of these dark cells is to secrete the hydrogen ions or bicarbonate ions. Juxtaglomerular apparatus, another interesting structure. So at the vascular pole, that is the region where the afferent renal arteriole enters into the Bowman's capsule. In the tunica media of afferent glomerular arteriole, smooth muscle cells are replaced by modified epithelial cells. The modified epithelial cells contain spherical nuclei and cytoplasmic granules, and this region or cells are known as juxtaglomerular cells, JG cells. So you can mention juxtaglomerular cells comprises of spherical nuclei and cytoplasmic granules, and these are the modified epithelial cells. So in the adjacent distal convoluted tubule, the cells that border the JG cells are narrow and more columnar. This darker and more compact arrangement of cells is known as macula densa. So the juxtaglomerular cells along with macula densa composes the rich structure which is called as juxtaglomerular apparatus. So this contains membrane bound secretory granules, I told you, of the hormone renin and helps to maintain blood pressure in kidney for the process of glomerular filtration to occur, which is the first step in urine formation. In fact, its cells act as baroreceptors and chemoreceptors too. So a small information once again. The nephrons as such are of two types. They are present in the cortex. They are called as cortical nephrons, which represent 85% of the nephrons in the kidney, while the second type is called as the juxtamedullary nephrons, which forms only 15% of the nephron. Juxta means near to. So the cortex medullary junction, juxtaglomerular nephrons are found in the junction of cortex and the medulla. And these nephrons, that is the juxtamedullary nephrons, play an important role in the kidney's ability to produce concentrated urine. I think with this I will stop the explanation about the histological details of mammalian kidney. The next slide is that of the references. You just go through the reference if you get the textbook and understand the lesson better. Fine. Thank you.